Again, thanks for the invitation. I'm really happy and excited to be here and talk to small non-coding RNA fans. And I will give really a brief introduction about our research. So currently we concentrate on three main research domains: computational biology of microRNAs with a focus on interactions with targets. And this will be the topic of my talk today. Small RNA interactions in bacteria that Shani will present after me. And we are also part of an Israeli consortium, CRISPR-IL. So as part of this consortium, we develop computational tools for genome editing with the CRISPR technology. So today I plan to talk about microRNAs, which I have been studying since being a PhD student. I will first give a brief introduction about the identification of microRNA targets, both by experimental and computational approaches. Then I will focus on the challenges involved in the application of machine learning models to microRNA target prediction. And finally, I will show how we have been using classic machine learning and deep learning approaches to investigate whether microRNA target interactions are transferable between species, or in other words, are the rules conserved throughout evolution. So I don't think microRNAs need much introduction in this audience. So just briefly, we know they regulate gene expression, and they are generated in a multi-step process from a long transcript all the way to a short mature sequence that is loaded into the argonaute protein, which then serves like a search engine. It scans messenger RNA triprime UTRs and binds those that have complementary sequences to microRNAs. And binding of the complex to these targets leads to translational inhibition or degradation. And we know that each specific microRNA can target hundreds to thousands different genes. And so far, microRNAs were shown to play key roles in diverse biological processes, including development, and they were implicated in various human diseases. So one of the important things in microRNA research is to identify their target genes. And why do we need that? So this is in order to understand what biological processes they are involved in and how can we do it. So there are experimental and computational approaches. And what exactly do we want to know? Well, this depends on the research question. On the lowest resolution, we just want to know what are the target genes. But if we want a higher resolution, we would also want to know the exact binding sites on the mRNA of the target genes. So there are several experimental high throughput methods for target identification. And the most common approach is microRNA overexpression or inhibition in cells, followed by gene expression profiling by methods like microarrays, RNA-seq, ribosomal profiling, etc. And the purpose is to identify genes whose expression changed due to change in microRNA levels. And though this method is easy and pretty straightforward, its biggest limitation is that it's difficult to distinguish direct and indirect microRNA targets, and there is also no information about the exact binding site. The second approach is cross-linking and immunoprecipitation of ago-RNA complexes, also known as ago-clip. This method recovers a pool of really precise binding sites on messenger RNAs and a pool of microRNAs. However, it's unknown which microRNA binds each site, and it has to be predicted bioinformatically. And the last method I want to mention is known as CLASH, or it has also some variation, cross-linking, ligation, and sequencing of hybrids. It was first introduced in 2013. So this method is very similar to ago-clip, but it has an additional crucial step in which a ligase is added to induce ligation between the microRNA shown here in blue and the target mRNA shown here in black. So if we, after we perform this protocol, if we look on the sequenced library, we observed 3 types of sequences. So we see chimeric fragments that contain both the microRNA and the target. We see target RNAs separately, and we see microRNAs separately. And the later 2 groups are also what we recover in a regular clip. So of those 3 groups, only the chimeric reads gives us precise information about interacting microRNAs and targets. But unfortunately, since the ligation step is not very efficient, the proportion of chimeric reads is very low. It's about 2% from total reads. And depends on the data set, this captures about hundreds to maybe thousands interacting microRNA target pairs. So looks promising, but is it enough? So this method captures only a small subset of all possible interactions. First, because of the low yield, and second, because each experiment is done on a very specific cell type or specific developmental stage. So we don't see all possible interacting partners. And in addition, because of the technical challenges of the method, there is data only for a few species. So as you can see in this tree of model organisms, data sets of chimeric microRNA target interactions, indicated here with the star, were generated only for 4 species: human, mouse, cow, and the nematode C. elegans. And altogether, there are only 8 data sets. So what can we do in other organisms where no experimental data is available? So here, computational prediction comes to the rescue in order to close the gaps in the experimental data. And the simplest task we can think of is, given a microRNA and a target gene or its messenger RNA, we want to compute whether they will interact, and if they do, where is the binding site? So to be able to do the prediction, we first need to understand what characterizes an interaction between a microRNA and its target. And these characteristics, also known as features, can be grouped into several main categories. So the first category is the sequence composition of the microRNA and of the target. And this can help, for example, to understand if there is a preference for specific nucleotides on the target. The second group contains features related to the target itself, such as the evolutionary conservation of the target gene, its accessibility, the sequences surrounding the target site, and more. And the third group contains features related to the duplex formed between the 2 sequences, okay? And it includes features like the strength of the duplex, which is reflected in the free energy that is released when they bind, or specific base pairing patterns, such as the binding in the seed region of the microRNA, and more. And so far, many methods were developed for this task. The first generation of tools was based on very basic rules of thumb. For example, free energy is lower than some threshold, perfect seed match at positions 2 to 8, up to 3 GU pairs in the duplex, etc. But since the interactions are very complex, it is really hard to cover all possible aspects using simple rules. So the availability of the high throughput datasets that I just told you about of direct microRNA target interactions led to the development of machine learning classifiers that are supposed to differentiate interacting pairs and non-interacting pairs. And to create a machine learning model, we need data. So in our case, we need pairs of microRNAs and genes, and we need a label for each pair that states whether this pair will interact. Is it a positive or not interacting, negative pair? And we need a representation of these pairs by some predefined features, for example, the features I just told you about. Then the data is split into 2 sets. The first set serves as a training set for the machine learning model to learn what differentiates between positive and negative pairs. And the second set is known as testing set, is then used to validate, okay, and test the performance of the model. And ideally, the model should be also tested on some independent test set that are not coming from the same data set. Then this model that we created is basically a prediction tool. It can take now new pairs represented by the same features and predict whether they will interact or not. So applying machine learning on microRNA target prediction requires to overcome some challenges and even points to consider. And I will now mention some of them. So first is the data itself. Okay, let's start with the interaction data, which is the positive data for our model. So in most cases, it requires pre-processing as there are no standards to report such data, and many times the details of the interactions are according to the generosity of the authors. Second is that the data may be biased because of differences in labs, protocols, cell types, etc. Third is, though it is believed that functional microRNA interactions occur mainly on 3' UTRs, a large portion of the reported interactions fall in other regions of the mRNAs. So should they all be taken into account, or we need to filter them out and keep only the 3' UTRs? In addition, it was shown that functional interactions should have at least partial match in the seed region of the microRNA. But we see a lot of seedless interactions as well. So should we throw or keep them? The negative data is very important to train a classifier, but there are no high throughput experimentally validated non-interacting negative pairs. So one of the approaches is to generate synthetic interactions of artificial microRNAs or mRNAs. But here we should be careful, since if the artificial sequences are too different or too similar to the real sequences, the machine learning method will not be efficiently trained. An additional approach is, for example, to infer negative interactions as anything that is not positive, but there are no standards. In addition, depending on the chosen negative data set, there could be imbalance between the two sets, which may cause underfitting or overfitting models. Features. Okay, I showed you that there are many features that could be considered: sequence, structures, accessibility, energy, and more. But what features to include and how to calculate them? So note that the experiment recovers the interacting sequences. So the microRNA and some mRNA fragment, but it doesn't really imply how exactly the nucleotides of those sequences interact. So features that characterize the hybrid that is formed between the two sequences have to be computationally predicted. And there are several programs that can be used for this task, but they sometimes compute different hybrids. For example, RNA hybrid and RNA duplex narrow down the long fragment to the fragment shown here in blue, but the base pairing pattern is different, as you can see here. While Miranda predicts a different region, here shown in orange, and different base pairing pattern. So again, what should we choose for a model? And there are, of course, more general issues as the machine learning method themselves. There are many methods. Each method has its own advantages and limitations. And also there are different evaluation methods. So all those points have to be considered. So what you see in this table is only a partial list of machine learning methods that were developed for microRNA target prediction. Here I focused only on methods that used clash-like datasets for training. And I, of course, do not expect you to read the content. I just want to point out that these methods mainly differ in the machine learning method that they use, the features that they use, the data that is used for training and testing, and the way they generate negative data. And here I want to get to the main point of our research. So most of the published papers on machine learning based target predictions were trained and tested on human microRNA target pairs. But we know that microRNAs are conserved and they are found in genomes of all animals. So one of the most important questions about this method refers to their general applicability and their ability to classify microRNA and mRNA pairs from organisms that are not human. And we actually used this question to try to answer a fundamental question in microRNA biology. So are the rules of microRNA interactions conserved across species? And the motivation behind this question is based on what I already told you. There are very few experimental data sets in very small number of species. So can we use models that we develop for this species that have training data to predict interactions in other species? And our approach to answer this question is to use machine learning to determine if and when we can adapt data and rules from one species to another. So our study was published a few months ago, so we'll just briefly touch the main things we found out and then show some very fresh results from our current work. Okay, so to address our research question, we developed the following flow. First, we retrieved all data sets of a chimeric microRNA target interactions. We processed them into a standard format and we filtered them based on some criteria. For example, we kept only interactions that fall on tripamide chiasm. Then we generated and evaluated machine learning model within the same dataset, just as a proof of concept. We also identified top influential features in each dataset. We then evaluated cross-dataset classification. And finally, we identified factors that could explain the results we got. And then I'll go now through the flow. Okay, so 8 datasets of cow, C. elegans, the nematode C. elegans, human and mouse, the datasets that I mentioned previously were processed in our study, and they contained between 600 to 18,000 interactions, as you can see here. So these are the positive interactions that are come from the experiment. So we generated also negative interactions using a synthetic method in which we shuffled the microRNA sequences in each interaction and then computed their interactions. And we used 490 different features to represent each interaction. And the features span different classes, a structure of the hybrid that we computed with RNA duplex, energy, accessibility, context, etc. So as a proof of concept, we first performed what we call intra-dataset classification. So we split each dataset to training and testing set. We build a model based on the training, and then we evaluate it based on the testing. And we use several metrics to evaluate the model. One of them is accuracy, which measures the fraction of correct predictions out of total. And the higher the accuracy, the better. And we also tested 6 different machine learning algorithms. And our results are summarized in this heat map. So each row is a data set, and each column is a different machine learning method. And in the cells, you see the accuracy level. So the accuracy ranged from 0.73 to 0.94. And generally, results above 0.8, 0.85 are considered high and significant. And as you can see, the method that is known as XGBoost achieved the highest results for all datasets. So we choose it as our main method for further analysis. We also analyzed top important features for each dataset and compared them. But due to lack of time, I will not show the results. I will just mention that we found a significant overlap between the sets, suggesting that there are common features that are influential among these datasets. Next, we evaluated our main question, which is cross-dataset predictions. In other words, we wanted to test what happens if we train a classifier on one dataset and then we test it on another dataset. Is it from the same or from the other species? And the reason we used 80% for training is because we just used exactly the same classifiers from the previous stage. So altogether, there are 56 possible combinations, considering each dataset either as a training or as a testing set. And this heat map shows for each pair of datasets the classification accuracy. So rows represent datasets that were used for training, and the columns are datasets that are used for testing. So what can we learn from this heat map? So first, if we look on the diagonal, it represents the intra-dataset accuracy, training and testing on the same dataset. And you can see that the results are variable. They are ranging from really random, slightly above 0.5 to all the way to 0.91. Pairs of data sets originating from the same species that are indicated by black boxes generally achieved higher accuracy than pairs from different species. You see it's more red colors inside the boxes, with few exceptions that I will discuss later. We also see that mixed pairs of C. elegans and other species had the lowest performance, shown here in blue. And finally, we can also see that the accuracy matrix is asymmetric. Here you can see an example. So training on a cow dataset and testing on human achieves a different performance than the swapped pair, training on human and predicting cow. So to explain these results, we examined several factors. So on the right, you can see the evolutionary history of the species in our analysis, including the time since the species diverged from their common ancestor. And the mouse and the human are the closest to each other, with cow equally and relatively close to them, and while C. elegans is the most distant from all. And indeed, the lowest accuracy was obtained for combinations of C. elegans with other species. So next, we measured for each pair of datasets how different they are based on their microRNA seed family distribution. And the motivation for that was that previous studies showed that different microRNAs may engage different base pairing patterns. And because of that, they may follow maybe different targeting rules. And a specific distribution of microRNA sequences in the training set may lead to biases in the rules learned by a machine learning model and maybe affect its performance on a testing set. So to measure the difference, we used a KL divergence, which measures information loss when we approximate a distribution P by some distribution Q. For example, when Q is a normal distribution and it approximates some experimental distribution. And in our case, we measure how much information is lost when we represent the testing set by a training set. So this heat map shows the divergence between each pair of datasets. Source datasets here in the rows are the one that we use for training, and target dataset is the one that is used for testing. The divergence of a dataset with itself on the diagonal is always 0, and the higher the value, the higher the divergence. Divergence between datasets within the same species, again, inside the black boxes, is typically lower than the divergence between different species. And again, the divergence between C. elegans and other species are significantly higher than the divergence between other pairs, which indicates that seeds distribution of other species poorly represent C. elegans datasets and vice versa. And finally, we also observed the asymmetry of the KL divergence. For example, these two human datasets. This means that H1, dataset H1 of human, better approximates dataset H3, and that information loss is smaller than the opposite case. Now, if we put both maps side by side, we can see that there is an agreement between the trends. Okay, note that the colors are inverse on purpose. Red is high accuracy and low divergence. So C. elegans with other species has higher divergence and low accuracy. And if we now focus on the two human data sets, H1 and H3 that I showed previously, where H1 better represents H3 than the opposite, we'll also see the classifier that is trained on H1 and tested on H3 performs better than the opposite. So KL divergence can explain low results that we see, for example, on this human data sets. On the other hand, if we now compare data sets H1 and H2 here, we see they have similar divergence in both directions, but the classifier that trained on H1 performs better than on H2 than the opposite. So KL divergence cannot explain all results. So finally, we used PCA, principal component analysis, to transform the interactions that are initially represented by 500 features into two-dimensional space. So every square is a data set and every dot inside is one interaction. So here we can see both the size and the spread of the data set. C. elegans is concentrated in a narrow area. Cow data set is big and widely spread. And in human and mouse, we see both widely spread and also concentrated data set. So the spread in this case indicates the diversity of the interactions. So the lower spread of H2 now can explain why a classifier that is trained on this has such low accuracy in predicting the interactions in other data sets. It is not diverse enough to learn from it. So to conclude this part, our findings suggest that target prediction models could also be applied to species for which experimental training data is limited or unavailable, as long as they are sufficiently close evolutionary to the species whose data is used for training and the training data is rich enough. So this was the published part. Now I want to show in the few more minutes I have the new results. So just summarize again, so far in our experiments, we used some source species to create a model, and we tested its performance on some other target species. But if we have now for this target data set, a small number of validated interactions, say 100 interactions. So training a model on this 100 interactions is not very useful. So we ask ourselves, can we use the model that we build for the source species and maybe enhance its performance on the target species by additional training with the available 100 interactions? So to test this question, we use the technique known as transfer learning. So in this method, we first train an artificial neural network on the source data set. We freeze the initial layers of the network, and then we retrain just part of the network with the new interactions. Then we test the performance of the transferred model. This is work in progress, and I want just to share some of our new results. So I also want to know that in our new experiments, we merged all data sets of the same species into one data set after we validated that it doesn't decrease the cross-species performance. So now we have 4 data sets altogether, 1 for each species. Then we split each data set again for 80% training and 20% testing. So first, let's see if we can enhance prediction from human to worm. The testing set in all these graphs is a worm data set. So the green line represents the results of a full training on worm and testing on worm, the best you can get if you have enough training data. The blue line represents training on small chunks of worm interactions between 100 to 500. And the orange line is in the middle represents the transfer from human to worm, where the 0 point is the regular cross, just training on human and predicting the worm, while the next points represent pre-training on human with additional training on worm chunks of 100 to 500 observation. And we see that a trend of the orange line increase in performance, and it's always above the blue line. So this suggests, and also it gets closer to the green line. So suggesting that we can improve performance using transfer learning in this case. And two more examples. Now let's look on the opposite direction. The testing is done on human this time. The green is a full train of human and testing on human, the best we can get for human. The blue line represents training on small chunks of human interaction. And again, orange is the cross results with transfer learning. The 0 point is pretty low, not just because the evolutionary distance between worm and human, but also because the worm dataset is relatively small. It's not very diverse. And we can see that both the orange and the blue line increase compared to the starting point, but they are really far from the green line. So, and seems that there are no much advantage to transfer learning compared to just training on small chunks of only human data set. And final example that I want to show is learning from cow to mouse. So here we test the mouse data set. Both data sets are pretty big. The green is training on full mouse data set. Cross from cattle to mouse performed pretty okay, but transfer learning provides very slight improvement. And training on small chunks of mouse interactions, 100 to 500, achieves much lower performance. So here we see the power of using data from close species, even with the slight enhancement of performance due to transfer learning. And as I said, these are fresh results, and we hope to finish analyzing them soon. So stay tuned if you are interested. And with that, I want to finish and really thank the students that worked and continue working on new directions. So Gilad performed the research on transferability of microRNA rules, and he developed all the pre-processing pipelines that we now enjoy to use. Eyal works on transfer learning techniques. Efrat and Liat examine directions related to negative data and targeting within different mRNA regions, And Shani, that will present her work shortly, tackles similar research question, but for interactions of bacterial small RNAs.